Hello, everyone, and welcome to today's webinar, Advanced Digital PCR Applications for Cell and Gene Therapy Research, Viral Vector Quantification and CAR T-Cell Monitoring. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. Due to time zone differences, Dr. Raquel Muñoz-Garcia will not be available for our live Q&A. However, any questions submitted for her, they will be answered via email to the email address you provided at the time of registration. Today's event will feature two talks. I would first like to introduce Dr. Raquel Muñoz-Garcia. Dr. Muñoz is an immunologist from Hospital Universitario Virgen de Rocio. Today, she will be speaking about the use of digital PCR for her research in CAR T therapy monitoring. Dr. Muñoz, welcome. You may now begin your presentation.
Hello, my name is Raquel Muñoz, and I'm immunologist at Hospital Universitario Virgen del Rocío in Sevilla. And I'm going to talk about CAR T-cell monitoring in clinical phase using digital PCR technologies. This is the script that I will follow during the presentation. First, I'm going to explain what the immunotherapy for cancer is, and I will focus on CAR T therapy, digital PCR, and how to monitoring CAR T therapy with digital PCR. First, the immunotherapy for cancer is based on the fact that the immune system has an important role in the development of cancer. The process of immunoediting or immunosurveillance is also called the 3S: elimination, equilibrium, and escape. And based on this important role of the immune system in cancer, many immunotherapy strategies for cancer have been developed, like immunocheckpoint inhibitors, classical T-cell transfer therapies, monoclonal antibodies, vaccines, immunomodulators, but I will focus on chimeric antigen receptors, CAR T-cell therapy. And what's the CAR T therapy is? Well, it's a therapy based on the capacity of chimeric antigen receptor T cells of bind a specific target of the cancer cells and kill them. The process consists on remove blood from a patient and isolates T cells. The rest of the blood is returned to the patients, and in the lab we make the CAR T receptor, and we insert this gene into the T cells and grow millions of these CAR T cells. Then we infuse CAR T cells into the patients, so these cells can find and bind to his specific antigen and kill the tumor cells. This is the schematic representation of a CAR T cell. There are three main domains: the extracellular domain, that is an antigen recognition domain that derives from an antibody and recognize specific targets and determine CAR T immunogenicity. The second domain is the transmembrane domain that is an spacer that provides stability and anchoring the CAR T receptor to the T cell. And finally, the intracellular domain, that is a signaling domain that facilitates T cell activation and provides persistence and survival of CAR T cells. This structure have been changed over the time from first generation to fifth generation, and the main difference between these generations are in the signaling domain. This domain have been improved by adding co-stimulators domains, and in its simplest version, it's also had the CD3 change of a TCR, and it has been adding other co-stimulators molecules. In the last figure, we can see the action mechanism of the CAR T receptor. The extracellular domain can recognize specific antigens of the tumor cells, and the activation signal go through the intracellular domain and activates the CAR T cell that kill the tumor cell in a specific way. These are the 6 CAR T cell therapies that have been approved by the Food and Drug Administration until 2016. And all these therapies have been approved for the treatment of blood cancers, like lymphomas, sometimes of leukemia, and more recently, multiple myeloma. This therapy can lead very good results in some patients, but like all therapies, it can also have second unwanted effects like inflammation, anaphylaxis, and more severe neurotoxicity or CRS, cytokine release syndrome. And in some cases, it can be severe or even fatal. So, CAR T therapy have demonstrated remarkable clinical results, but also failures and serious side effects. And what it depends on? To answer this question, we need some biomarkers, and peak expansion and persistence of CAR T are correlated with responses rates and severity of side effects. So, in vivo CAR T cell expansion following have been recognized as an important surrogate marker for response and disease control. But no standardized method or commercial safe for CAR T measurement is established yet. For this reason, flow cytometry or quantitative real-time PCR have been proposed to monitoring CAR T therapy, but both techniques have many disadvantages that no facilitate their introduction in the clinical practice. So we thought, and what about digital PCR? So in digital PCR, the K is the partition, and in real-time PCR, the accumulation of amplification product is measured as the reaction progresses after each cycle. So there is competition of the reagents, and in cases of low-frequency target, it is possible to lose the amplification and only amplificate the high-frequency target. But in digital PCR, thanks to partition, we isolate DNA molecules in each compartment. So the detection limit is the same as the quantitative real-time PCR, but the reproducibility is higher, and this is a great advantage for low-frequency targets. One of the great disadvantages of the old platforms of digital PCR was that the significant data volume until a health, limited or inconsistent compartmentalization, a tedious workflow. Different devices were used, and a longer waiting time for results until 9 hours. But with the new applied biosystem Quant Studio Absolute Q Digital PCR, the dead volume is less than 5%. We had about 20,000 microreaction, and the workflow is super easy. And we obtained the results very fast, in an hour and a half. So we thought about monitoring CAR-T therapy by digital PCR. We use samples of 46 patients with relapsed or refractory acute lymphoblastic leukemia or lymphoma B, treated with CD19 CAR-T, tisa-genlecleucel, or axicabtagen-thiloleucel. And we use samples in different time points after infusion of the CAR-T, days 5, 7, 11, 14, 20 or 23, and 28 and 30. And we obtain genomic DNA from peripheral blood mononuclear cells of each sample. The experimental design consists in the simultaneous amplification of two targets. In the one hand, the FMC63 fragment present in both CAR-Ts, and on the other hand, the amplification of RNase P present in all cells. So we used specific primers and approved labeled with fluorophore to amplify the FMC63 present only in the CAR-T construction. And we use a specific set, primer plus proof, labeled with big fluorophore to amplify RNase P present, as I said before, in the CAR-T, but also in the other cells. So the workflow is isolate DNA samples, prepare the PCR reaction, load the MAP16 plate, run the absolute Q, and then analyze data. This is a real example of a patient. Here we have the figure that gives you the software. Here we can see the big fluorescence that shows the amplification of the RNase P gene. And here we have the fan fluorescence that shows the amplification of CAR-T gene. And the software gives you the copies per microliter of CAR-T and RNase P in each sample in day 5, 7, 11, 14, 22, and 30. And to normalize and compare this data between the different days, we applied this mathematical formula and obtained the CAR-T copies per cell. As I said before, we know that there are 2 copies of RNase P per cell, so we can calculate the number of cells that it will be equal of copies of RNase P between 2. So here we have the same data but normalized and expressed as the copies of CAR-T per cell. And considering all data of the patients, we can say that patients show expansion of the infused product followed by a rapid loss until day 20, after which detection is very low. And if we represent this data over the time in a graphic, we can distinguish 3 groups. Group 1 is the group whose expansion of the infused product occurs between days 10 and 11, and the great majority of patients are in this group, the 82% of patients. This is the typical expansion curve of this group. Group 2, whose expansion of the infused product occurred after day 11, there are only 4% of the patients in this group, and here we have the typical curve of the expansion of this group. And finally, the third group that presents no expansion of the infused product. Also, the 4% of the patients are in this group. So with these results, we can also say that are qualitatively similar to those obtained by flow cytometry with the same samples. And all of these results have been published in Frontiers of Immunology, and this work was made by the hematology group in my hospital, and we collaborated with our data. And at this point, I wanted to thanks, especially to the immunology group of my hospital, for giving me the opportunity. So what about the future? After 20 days post-infusion, the detection of CAR-T is very low, and we have some questions about this. Could digital PCR detect small amounts of CAR-T when flow cytometry cannot? Or could digital PCR be a tool with sufficient sensitivity to detect CAR-T after 20 post-infusion? So we are working to answer these questions. In summary, first, monitoring of CAR-T cells in vivo might be a fundamental tool for the identification of biomarkers associated with response or toxicity at very early time points post-infusion, which will serve to provide earlier interventions. Second, monitoring by Applied Biosystem QuantStudio Absolute Q digital PCR. Most of the patients show expansion of the infused product between days 10 and 11, followed by a rapid loss until day 20, after which detection is very low. These results are qualitatively the same as those obtained by flow cytometry. And finally, due to its simplicity and reproducibility, digital PCR could be useful in the quantification of CAR-T in follow-up and monitoring. So that's all, and thanks for your attention.
Thank you, Dr. Muñoz. Next, I would like to introduce Kim and Himani, who will be speaking about high-throughput viral vector quantification by digital PCR. Himani Patel is a scientist at Thermo Fisher Scientific, specializing in the development and release of dPCR and qPCR products for the cell and gene therapy field. With a background in chemistry and manufacturing, she brings expertise in research and development to advance molecular tools and technologies. Kimberly Gomez is a scientist at Thermo Fisher Scientific, specializing in the development and release of dPCR and qPCR products for the cell and gene therapy field. With a background in biochemistry and molecular biology research, she brings experience in analytical verification and clinical validation testing, as well as research and development for new products in the cell and gene therapy space. Welcome, Himani and Kim. You may now both begin your presentations.
Thank you, Christy, for the introduction. Hello, everyone. My name is Himani Patel, and I'm a molecular biology scientist here at Thermo Fisher. I'm also joined by Kimberly Gomez, also a scientist at Thermo Fisher. Today, I'm thrilled to have the opportunity to share advancements in automating digital PCR workflows, particularly in the context of achieving consistent AAV quantification using the QuantStudio Absolute Q auto-run dPCR suite. Over the next 30 minutes, I will discuss the research avenues for cell and gene therapy and provide a brief overview of digital PCR technology and our Absolute Q auto-run dPCR suite. Kim will then dive deeper into strategies for gene therapy research using AAV vectors, considerations to take with AAV sample preparation, and share data that demonstrates consistency of AAV quantification using the auto-run. Cell and gene therapy research is highly impactful due to its potential to revolutionize the field of medicine and significantly influence human health. Research in this field offers the possibility of treating or even curing rare or challenging genetic disorders or diseases. This may help patients suffering from conditions such as cystic fibrosis, muscular dystrophy, and sickle cell anemia. Cell and gene therapy research also helps to drive innovation towards the development of new therapeutic modalities and advanced technologies. The rapid progress in gene editing, such as CRISPR-Cas9, has opened up unprecedented opportunities for precise and efficient genome manipulation. Additionally, stem cell therapies hold immense potential for regenerative medicine and tissue engineering. Lastly, cell and gene therapy research helps advance our understanding of disease mechanisms, cellular biology, and genetic regulation. By studying the interactions between genes, proteins, and signaling pathways, we gain valuable insights into disease progression and the underlying biological processes. This knowledge serves as a foundation for the development of targeted therapies and precision medicine. Cell therapy targets living cells to remove, modify, and reintroduce to a patient, and these living cells can be sourced from the patients themselves or from donors. Cell therapy research helps us better understand cell types, properties, and applications. For example, research in this field helps us investigate stem cells and explore methods to deliver cells to target tissues. Gene therapy, on the other hand, modifies genetic material to introduce to a patient's cells. In an ex vivo approach, cells are taken from a patient's body, modified, and then reintroduced. In an in vivo approach, the modification of the genetic material is performed within a patient's body. The altered genetic material may be used to investigate the source of genetic defects. Gene therapy research helps us understand the underlying causes of diseases, investigate techniques to regulate gene expression, and helps us explore and discover gene delivery vehicles. To deliver high-quality cell and gene therapy products, stringent analytical evaluation is critical. In gene therapy, characterization tests such as confirming plasmid or vector constructs or confirming the modification of a gene is conducted. Manufacturing tests such as monitoring the quantification of a viral vector or gene copy number and verifying the packing efficiency of AAV titers is administered. In cell therapy, characterization tests also include confirming vector constructs and gene editing events. Vector titers are also tested as part of the manufacturing process. Testing for impurities such as residual DNA or contaminants is also critical to product quality in both gene and cell therapy. Digital PCR is a valuable tool to perform these various analytical tests. So what exactly is digital PCR? Digital PCR is a method of quantifying nucleic acid targets without standard curves by dividing the bulk reaction into thousands of smaller independent reactions. PCR at large can be organized into three major categories: bulk PCR, quantitative PCR, and digital PCR. Bulk PCR leverages endpoint data to provide a yes or no answer. Is the target of interest present or not? Quantitative PCR provides real-time measurements and requires the use of a standard curve to provide relative quantitative data. Digital PCR does not require a standard curve and is capable of providing absolute quantification of known genetic targets. Because no standard curve is required, this method is considered to offer greater precision and reproducibility when compared to other quantitative methods. Like I mentioned in the previous slide, digital PCR divides a qPCR reaction into thousands of smaller independent reactions. The reactions are segregated into microchambers, where individual molecules are then amplified. The presence of the target molecule is simply counted in each microchamber. Digital PCR not only circumvents the need for a standard curve, but it also allows for target analysis in low quantities. It uses highly precise and specific technology that can monitor small changes over time and is resistant to PCR inhibitors. After compartmentalization and PCR is performed, individual compartments or microchambers that contain at least one target molecule prior to amplification will fluoresce and will be easily distinguishable from dark microchambers, which do not contain a target molecule prior to amplification. The total number of fluorescent microchambers are counted and used to calculate the absolute quantity of the target within the sample. On the left, we see a fixed digital PCR array post-amplification consisting of approximately 20,000 individual microchambers. On the right, we see a typical one-dimensional scatter plot, a commonly used graphical display of digital PCR data. Each dot on this plot represents the relative fluorescence of a single microchamber. All dots located above the threshold are considered positive for the target, while those below are considered negative. This information is then used to calculate the absolute concentration of the target. The absolute Q is a single instrument which consolidates all three major functions of digital PCR: reagent manipulation, amplification, and data acquisition, into a single box. It doesn't require a standard curve, enables molecular integrity analysis, and is an excellent tool to detect low concentrations or rare targets. It's incredibly easy to use, requiring only a single consumable and less than 5 minutes of hands-on time. The typical runtime for a full plate is approximately 90 minutes, which includes compartmentalization, PCR, and data acquisition. More than 95% of a loaded reaction is consistently divided into 20,000 plus fixed microchambers. Plates are designed to allow for flexibility, allowing for 4, 8, 12, or 16 samples to be run in parallel, while unused arrays can be preserved for use later. The absolute Q also includes 4 optical channels for increased multiplexing capacity, with an added 5th channel devoted to a passive reference dye. Additionally, the absolute Q images each fixed array both before and after PCR is completed. This allows for the subtraction of any pre-PCR fluorescence prior to analysis. Overall, the absolute Q delivers high-quality data and easy-to-use workflow with flexibility to meet laboratory testing needs. So to summarize, it's important to consider conducting nucleic acid measurements that avoid the use of standard curves, provide consistent data between batches and concentration ranges, and have the ability to accurately identify and measure the presence of single molecules. Workflows should also be scalable for both low and high throughput sample processing. So on that note, I'd like to hand it over to Kim to talk about Thermo Fisher's QuantStudio Absolute Q auto-run instrument and technology and how it demonstrates consistency in AAV quantification.
Thanks, Himani. AAV, or adeno-associated virus, is a viral vector typically used in gene therapy. AAVs are traditionally made by performing triple transfection using 3 plasmids. One would be a transferred plasmid that contains your gene of interest, or your transgene, and that transgene is flanked by 2 ITR regions. A second plasmid would contain rep and cap genes to make your rep and cap proteins, and a helper plasmid that contains genes for mediating AAV replication in the packaging cell line. AAV is then harvested from the packaging cell, purified, characterized, and eventually administered to people as a therapeutic. There are multiple critical quality attributes, or CQAs, that impact the final AAV vector product. Measuring these CQAs may or may not require an analytical control. One attribute would be gene expression, or looking at genome integrity. Was the transgene incorporated into the plasmid? And did the AAV receive the full intact DNA? Another would be looking at packing efficiency. Was the DNA encapsulated into the AAV capsid? Another is identity and potency of the AAV. Is the correct AAV serotype used? And what is the infectivity of AAV? Fourth, we could look at vector purity. How sterile is the AAV product? Are there trace amounts of contaminants like mycoplasma or residual DNA from the host cell? Lastly, we can look at vector titer, or looking at the concentration of AAV, which is important for potency and dosing of the final AAV product. All of these attributes are important for AAV, but the one that we're going to narrow in on in this presentation is AAV vector titering. With vector titering, your method of measurement needs to be consistent and precise. You wouldn't want to put too much or too little of your therapeutic into the patient. In this study, to demonstrate consistency and precision on the Absolute Q platform, we measure recombinant AAV genomic DNA along with a control using primers and probes that target the ITR2 region. ITR2 is an important gene for AAV packaging. It's required for the proper translocation of genomic DNA into the AAV capsid. So for our study, we used primers and probes from Applied Biosystems, which target the ITR region. For input DNA, we have two types of samples. One is AAV particles from Vigene. These are AAV2 serotype, and these particles contain an EGFP transgene. Our second sample is double-stranded DNA control that contains the same sequences in the Vigene particles. Before being tested on the Absolute Q, the viral particles go through a short sample prep workflow. Starting with the stock samples on the left of the diagram, the AAV particles are diluted in a low TE buffer containing 0.01% pluronic F68. This is a surfactant that helps with preventing viral clumping and with preventing sticking to plastic walls. Next, the viral particles undergo heat lysis on a thermal cycler at 95 degrees Celsius for 5 minutes. This helps with breaking open the capsid and releasing that DNA for testing. This sample is then mixed with our master mix and our ITR assay. Then the sample is loaded on the MAP16 plate. This plate can then be directly placed in our Absolute Q instrument or can be placed using the auto-run suite. The Absolute Q auto-run dPCR suite uses Thermo Fisher's Spinnaker microplate robot. With this suite, we're not only maximizing sample throughput, but also data throughput by enabling multi-plate and multi-batch analysis. We have true walk-away automation because of the stability of our DNA dPCR reactions. We can provide 72 hours of hands-free walk-away automation, so you can do things overnight or over the weekend with confidence. With improvements in software, our instruments are lab automation compatible, and we have a menu of options to help with your laboratory needs and to enable greater productivity. We have a menu of options for dPCR users to choose from. You can start your process development with our Absolute Q instrument and then scale up with AutoRun. With the AutoRun suite, we allow for either a single or dual configuration. And this table summarizes all the features for each of these setups. I won't go into all of the details, but our upgradable options allow for lots of flexibility and scalability. So, using AutoRun for this study, we aimed to demonstrate consistency and precision on the Absolute Q. Our study consists of 20 MAP16 plates, divided evenly for testing samples at a single concentration, and then the other half to test a 5-point dilution series. And these 10 plates are further divided between our two sample types, either the AAV particles or our DNA control. Our analysis parameters for this study were based off of acceptance criteria typically used for evaluating AAV titers. So the two parameters that we're looking at are percent recovery of 80 to 120%, and then also percent CV of less than or equal to 15% for at least 6 replicates. Looking at our results, we'll start with the left graph, where we are looking at our ITR concentration of AAV particles across 5 plates. Our samples here are tested at 1 concentration. As you can see, our data points fall well within that 80 to 120 range of percent recovery, and our percent CVs are low. The highest percent CV for 1 plate is 3%. So we fall well below the 15% acceptance criteria. And the same thing could be said for our DNA control in the graph on the right in terms of percent recovery and percent CV. We also went a step further, and we did a one-way ANOVA test with all of our results and found that there's no significant difference in ITR copies between our MAP16 plates. In the second half of our study, we used a 5-point dilution series rather than one single concentration. As you can see in the graph on the left, we evaluate the dilutional linearity of our samples on the absolute Q. Our R-squared value here is 0.995, which indicate a strong linear relationship between our expected copies and our actual results. In the graph on the right, we normalize our results by back calculating to the stock concentration of the sample. So we do this by using the dilution factors needed to create the 5-point dilution series. Here you can see that, again, our results fall within that 80 to 120% recovery range, and our percent CV is, again, very low. Our highest percent CV for one plate is 3.2%. So again, well under that 15% CV acceptance criteria. To conclude, we have shown you the benefits of dPCR technology. You don't need a standard curve. Our workflow is scalable to your laboratory needs. We didn't discuss this too much here in the webinar, but with Absolute Q, we do enable molecular integrity analysis when using multiplex assays. And we have fast time to results of about 90 minutes to your first answer. Our study has shown consistency across multiple plates with low percent CV. We demonstrate precision with strong linearity across 5 dilution points. And we also demonstrate accuracy with all of our sample replicates, falling within that 20% recovery range. With this, we showed that the Absolute Q delivers high-quality data and easy-to-use workflow with flexibility to meet your laboratory testing needs. I want to thank you for your attention during this webinar, and we're open for questions.
Thank you, Dr. Muñoz and Kim and Himani, for your excellent presentations. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have any questions you would like to ask, please do so now. Just type them into the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, now let's get started. Our first question is, can we leave samples in the auto run suite overnight without loss in data quality?
So I can speak to that. We've done benchtop stability with our Absolute Q instrument, at least in terms of AAV samples. When after they're lysed and they're sitting in the reaction mix, we don't see any loss of data quality for, I think, 96 hours. So you can definitely leave it overnight, even over the weekend if you really needed to.
Thank you, Kim. Our next question. Do I need to decide on the high throughput system up front, or can I start with a lower throughput system first?
Yeah, so you can definitely start with a lower throughput system first. If you needed to do your process development, you could start with one instrument. And then when you're ready to scale up, you can choose to look into the auto run suite with either the single or the dual configuration. So it's definitely flexible to whatever needs you have in your laboratory.
And Kim, have you compared your results to results generated on the other dPCR platforms?
I have. So without naming names, I could say that the Absolute Q does perform just as well as the other dPCR platforms that are out there, especially for AAV quantification. Results are comparable. The percent CVs and the percent recovery that we showed in this presentation, they're also comparable with the other instruments as well.
Thank you. And it looks like we have time for one more question. What are the main advantages of using digital PCR in gene therapy research?
So a lot of the main advantage for digital PCR would be having to not use a standard curve. It's still complementary to existing qPCR workflows, but dPCR does provide a high precision and absolute quantification, again, without the need for a standard curve. And as I kind of hinted earlier in the webinar, there's the ability to look at single molecules with dPCR. So when you're doing a multiplex assay, you can look at a single DNA strand and look at that maybe like a gene of interest compared to ITR, and then look at the integrity of that molecule.
Thank you. Now, I'd like to thank Dr. Raquel Muñoz-Garcia, Himani Patel, and Kimberly Gomez for their time today. I would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go, I do want to thank the audience for joining us today and for their interesting questions. Questions we were unable to answer today and those submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope you have a great day.